Hello, welcome to Bite Size Med. This video is on the hypothalamic pituitary thyroid axis. The thyroid gland synthesizes and secretes the thyroid hormones, that's triiodothyronin and thyroxine, T3 and T4. It does that because it's being told to do it by the anterior pituitary, which in turn gets influenced by the hypothalamus. Together, these three form the hypothalamic pituitary thyroid axis. And we're going to start at the top with the hypothalamus. The hypothalamus in the brain produces a thyrotropin releasing hormone. That's TRH for short. Now, I know terms like this can get confusing, but at the end of the video, I'll try and help you out with them. Bear with me for now. This is a tripeptide derived from 3 amino acids: glutamic acid, histidine, and proline. And this TRH comes from the hypothalamus. Now, the hypothalamus and the pituitary are connected. Their vascular connection is what's important here. TRH has to reach the anterior pituitary, and does that because of these vessels. The hypothalamus has a capillary plexus, and hormones synthesized enter these capillaries. The capillaries then form veins, portal veins, since they're going to connect this capillary plexus to another around the anterior pituitary. Through this portal system, the hypothalamus can control the anterior pituitary, which is also known as the hypophysis. This is the hypothalamic hypophyseal portal system. The importance here is that the portal blood has TRH, since the hypothalamus synthesizes it, and the site that receives the highest concentration of that TRH is the anterior pituitary. The anterior pituitary synthesizes quite a few hormones, so it has cells for that process. The ones we need here are called the thyrotrophs. They're also known as thyrotropes or thyrotropic cells. TRH has receptors on these cells. This receptor is coupled to a G protein, which activates phospholipase C when TRH binds to its receptor. That catalyzes the breakdown of phosphatidylinositol 4,5-bisphosphate into diacylglycerol, that's DHE, and inositol triphosphate, that's IP3. DAG activates protein kinase C and phosphorylates proteins. IP3 mobilizes calcium out of the intracellular stores, increasing the intracellular calcium. Eventually, this results in the synthesis and release of a hormone stored in the secretory granules of the thyrotrophs. That is thyrotropin, or the thyroid-stimulating hormone TSH. So TRH comes from the hypothalamus, and TSH comes from the anterior pituitary. TSH is a glycoprotein. It shares structural similarities to the gonadotropins. That's the luteinizing hormone, LH, and the follicle-stimulating hormone, FSH. It has alpha and beta chains. The alpha chains are common to all these hormones. It's the beta chains that make them specific. But once TSH is released, it's going to do exactly what the name suggests. It's going to stimulate the thyroid gland. So it has TSH receptors on the thyroid gland. These receptors are located on the thyroid follicular cells, also called thyrocytes. Note how they sound similar to thyrotropes, but thyrotropes are in the anterior pituitary, thyrocytes are in the thyroid gland. Now we're talking about TSH receptors on the thyrocytes. Again, it's a G protein coupled receptor, but a different G protein here, different second messenger as well. With TRH, it was DHE and IP3 that worked as second messengers. Here, with TSH, it's cyclic AMP. The G protein activates adenylyl cyclase when TSH binds to its receptor. That converts ATP to cyclic AMP. This activates protein kinase A and causes protein phosphorylation. Thus, TSH uses cyclic AMP as a second messenger. The effect of that is it stimulates almost every step of thyroid hormone synthesis. Though I covered most of this stuff in part 3, let's quickly review the steps of thyroid hormone synthesis. It needs iodine and tyrosine. The cell takes up iodine from the plasma by a sodium iodide symporter. That iodide ion then reaches the follicular lumen where it gets oxidized to iodine, so that thyroglobulin can be iodinated with the help of thyroid peroxidase. That forms mono and diiodotyrosine, which stay bound to thyroglobulin. This is organification of thyroglobulin, which happens in the follicular lumen. Then they get coupled, forming T3 and T4. This thyroglobulin, with all its attachments, gets endocytosed into the follicular cell. It undergoes proteolysis, releasing T3 and T4 so that they can get secreted into the bloodstream and systemic circulation. The rest gets recycled with a deiodinase to form iodine and tyrosine, which are used for the next cycle. TSH stimulates all these steps: the iodide uptake, the organification, the coupling, the endocytosis, the proteolysis, and secretion of T3 and T4. It's when the thyroid gland gets stimulated by TSH that the endocytosis of colloid from the follicular lumen with those hormones happens at all. The earliest effects of TSH would be on the last few steps, the proteolysis and release. These are the immediate actions. The effects on the rest take longer. The intermediate actions of TSH would involve protein synthesis. What kind of proteins? The sodium iodide symporter, thyroid peroxidase, etc. It can also have more long-term actions. In excess for a long duration of time, it can cause hyperplasia and hypertrophy of the thyroid follicular cells. Hyperplasia means the number of cells increases. Hypertrophy means it makes them larger. That would make the gland itself larger. They also increase capillary proliferation and blood flow to the gland. So in excess for a long time, TSH can make the thyroid gland larger, sometimes large enough to be visible as a swelling in the neck, and that's called a goiter. Now, a goiter only refers to the enlarged size of the gland. One can't draw conclusions about the thyroid status just from the presence of a goiter. When the thyroid gland secretes normal amounts of hormones, that's considered euthyroid. When a lesser amount is released, that's hypothyroid, and more is hyperthyroid. But a goiter can happen in any of these conditions, so it doesn't actually tell us about the thyroid hormone status. It just tells us that the thyroid gland is enlarged. And one cause of a goiter is the effect of too much TSH on the gland. But physiologically, at normal levels, it doesn't do that. It stimulates the normal release of T3 and T4. Those are our thyroid hormones. Now, most of what's secreted by the gland is T4, but T3 is the one that's more active. So in the periphery, T4 gets metabolized to T3 in organs like the liver, kidney, and skeletal muscle. That's by a deiodinase. It also forms reverse T3 in this process, which isn't supposed to have biological functions. T4 and T3 travel in blood, either free or bound, but mostly bound. However, it's the free form that's actually physiologically active. The thyroid hormones, mainly T3, reach the target cells and act on the nucleus of the cell. They bind to the thyroid hormone receptor, which forms a heterodimer with the retinoid X receptor, and this complex binds to the thyroid response element of the cell's DNA. This is followed by transcription and translation, so protein synthesis. And those proteins are how the thyroid hormones have effects on different parts of the body. So TRH from the hypothalamus, TSH from the anterior pituitary, and the thyroid gland forms T3 and T4. This axis is important for homeostasis. If there's too much thyroid hormones, that can result in hyperthyroidism or thyrotoxicosis. If there's too little, that's hypothyroidism. Now, those are pathological, and we don't want that. So how does the body regulate this? By feedback. When there's too much T3 and T4, there's negative feedback on the anterior pituitary. That's the short loop. There's also negative feedback on the hypothalamus. That's the long loop. Now, these structures know that the thyroid hormones are high. So what will they do? Reduce their hormones. So TRH and TSH come down. And when TSH comes down, it reduces the formation and release of T3 and T4, bringing their levels back to normal. If the thyroid hormone levels are too low, again feedback on the higher structures and the opposite will happen. Since the hormone levels are low, they need to bring it back up. TRH and TSH increase, eventually bringing the thyroid hormone levels back up to normal. How do they make all of this happen? The thyrotrophs of the anterior pituitary are sensors. They're mainly checking for T3. T3 enters either from the plasma or it can form inside the thyrotrophs directly. That's because the anterior pituitary has another type of deiodinase, the type 2 deiodinase, which has a high affinity for T4. It can convert T4 to T3, sort of reflecting the circulation levels of T3. So this intracellular T3 exerts negative feedback on the anterior pituitary. If the free T3 and T4 are high, they can either directly inhibit the synthesis of alpha and beta chains of TSH, or they can work indirectly by downregulating the TRH receptors on those thyrotrophs. That would reduce their sensitivity to TRH coming from the hypothalamus and thus indirectly lower TSH. So that's on the anterior pituitary. But what about the hypothalamus? By negative feedback, T3 can decrease the expression of pre-pro-TRH, lowering pro-TRH, reducing TRH, which will then decrease TSH, and thus lower the release of the thyroid hormones when they're high. Now that's just a general overview of this axis, but once you've understood how it works, the next step is getting to know what happens in different pathological conditions, hyper and hypothyroidism, because the levels of these hormones will obviously change, and the careful assessment of the levels of these hormones in relation to each other helps in understanding the status of thyroid gland function. Now I'm going to help you remember some of the terms in this axis. First, look at the letters and remember the alphabet. They're in order. So R, S, T, TRH, TSH, T3, and T4. So if you quickly need to remember where TRH comes from, it's the first, so it's the hypothalamus. But if you remember the expansion of each, which is preferable, it does make sense. So we have the hypothalamus, the anterior pituitary, and the thyroid gland. The thyroid gland produces triiodothyronine and thyroxine, T3 and T4. Triiodo. Tri is 3, so T3, that's easy, and that makes the other one T4, thyroxine. The cells of the thyroid gland are the thyrocytes, or thyroid follicular cells. Makes sense since they are cells of the thyroid gland, so thyrocytes. TSH is the thyroid stimulating hormone. It literally stimulates the thyroid gland directly, so it's from the anterior pituitary, and it's produced by thyrotrophs. Troph means nourishment and food and growth. So what feeds the thyroid gland? TSH from the anterior pituitary, which is also known as thyrotropin. So the cells that create that TSH are thyrotrophs or thyrotropic cells or thyrotropes. The hypothalamus is the one that tells the anterior pituitary to release the hormone. So it's a thyrotropin, not thyroid, thyrotropin releasing hormone, TRH. I hope that was a little helpful with the terms. But all that was about the relationship between the hypothalamus, the pituitary, and the thyroid gland, the hypothalamic pituitary thyroid axis. I hope this video was helpful. If it was, you can give it a like and subscribe to my channel. Thanks for watching, and I'll see you in the next one.